Composition of gastric juice. Normal gastric juice is around 2.5 liters per day, and the pH is around 1. If you see the composition, 99.5% of is water and 0.5% is solids. Solids include both organic and inorganic components, where organic constituents they constitutes mainly the enzymes, that is pepsinogen, gastric lipase, and gelatinase. The mucus secreted is of two types: the thick insoluble mucus and thin soluble mucus. Thick mucus is mainly secreted by the surface epithelial cells, whereas thin, they are secreted by the neck mucus cells of the pyloric and cardiac glands. And also it contains intrinsic factor. If you see the inorganic components, they mainly made up of hydrochloric acids, sulfates, bicarbonates, sodium, and potassium. Some of the further details about the individual components, like HCl. HCl is necessary for conversion of pepsinogen to pepsin. It has a bacteriostatic action, that is, food contains many bacteria, but they are killed by the gastric HCl, so that the contents of the upper part of the small intestine are bacteriologically sterile. The hydrochloric acid also stimulates the secretion of the GI hormones, and it has a preventive role against the gastric cancer. Persons who suffer from achlorohydria are thus more susceptible to gastric cancer. Pepsinogen. In the presence of HCl, pepsin is very active and it digests the food protein. Thus, the combination of acid and pepsin is called as acid-pepsin mixture, that is APM, and it is a powerful digester of the protein. Mucus. Mucus has the following functions. Especially the thick mucus, it protects the gastric mucosa mechanically from being injured by the corrosive effects of food. And together with bicarbonate ions of the gastric juice, it constitutes the mucosal barrier, which prevents the autodigestion of the gastric mucosa itself by the acid-peptic mixture. The soluble mucus makes the food lubricated. Bicarbonates in them act together with the gastric mucus to prevent the autodigestion of the stomach. Then intrinsic factor is needed for the absorption of orally ingested vitamin B12, whereas gastric lipase converts triglycerides into fatty acids and glycerol, whereas gelatinase liquefies gelatin. So this is the protein contained in the connective tissue. Coming to the hydrochloric secretion, the acid formed in the intracellular canalicles of parietal cell, that is not within the cytoplasm, so that the cell is not damaged due to the high acidity. The intracellular canaliculi extend from apical surface of the parietal cell into the interior, and they contain hydrogen potassium ATPase and chloride channels on the walls. The basolateral membrane on parietal cell in contact with the interstitial fluid and contain chloride bicarbonate exchangers and sodium potassium ATPase. When parietal cells stimulated, the carbon dioxide and water combine to form bicarbonic acid, which splits into hydrogen and bicarbonic acid bicarbonates in the presence of enzyme carbonic anhydrase. The hydrogen is pumped into the lumen of canaliculi with the help of hydrogen potassium ATPase or proton pump, so which is the active transport. The bicarbonate leaves the parietal cells by an antipode in the basolateral membrane of parietal cells that exchange chloride for bicarbonic ions. The bicarbonate ions binds with the sodium to form sodium bicarbonate in the capillary blood. The chloride in the cytoplasm diffuse into the lumen of canaliculi. In the lumen of canaliculi, the H plus combines with the chloride to form hydrochloric acid. This hydrochloric acid reaches the gastric lumen. So this is the diagram showing the production of HCl in the parietal cell, the activated parietal cells. So because of the metabolism, carbon dioxide is produced that combines with water in presence of carbonic anhydrase to form the bicarbonic acid, which forms the H plus ion and bicarbonate ions. So the H plus ions, they are apically, they are secreted into the canaliculus with the help of hydrogen potassium pump. And at the basolateral membrane, the bicarbonates are exchanged with the chloride ions. So the chloride ions, when they are increased intracellularly, they are going to diffuse into the canaliculi, and that is going to combine with H plus ion to going to form the hydrochloric acid, which is secreted into the stomach. At the same time, when the potassium concentration increases in the cell, so that will be exchanged with sodium potassium pump, and when the sodium enters inside the cell, it is going to drag some amount of water also, so which is going to diffuse into the canaliculi, which will be secreted with the hydrochloric acid. For each molecule of HCl formed in the gastric lumen, one molecule of sodium bicarbonate is formed in the blood. Hence, when HCl secretion is increased after meal, the pH of the blood increases, and this is called as postprandial alkaline tide. Some of the inhibitors of HCl over the parietal cell are their somatostatin, the acid chyme. It can have a direct action which inhibits the parietal cells directly, and indirect action will be that it increases the secretion of or production of secretin. Fatty food is going to inhibit the HCl secretion by a release of GIP. So this inhibits the gastric G cells and the parietal cells. Whereas enterogastrone, which is released because of the fatty acid, it is going to inhibit the HCl action of HCl. Then hyperosmotic chyme, which is going to inhibit the secretion and motility reflexly, and prostaglandins, particularly prostaglandin E, is going to inhibit